The pancreas and liver contribute secretions to the GI tract that are important for the digestive process. So the pancreas is a large gland and it has both exocrine and endocrine components. So the exocrine component makes up most of the pancreas, and it consists of closely packed secretory acini which drain into a branched ductal system. The exocrine pancreas is arranged kind of like a bunch of grapes, where each grape is a single acinus and the stems are the ducts. We saw the same type of organization in the salivary glands. So the acinus is lined with these acinar cells that store and secrete digestive enzymes, while the centroacinar cells and the ductal cells secrete electrolyte component of pancreatic juice. It's these combined secretions of enzyme and electrolyte and water that are carried through the ductal system to the main pancreatic duct eventually, all the way through to the common bile duct, through the sphincter of Oddi and into the duodenum. The final secretion of the exocrine pancreas is really high in enzyme as well as bicarbonate, so it is alkaline. The endocrine portion of the pancreas, which you can see an example of here, is made up of the islets of Langerhans, and they secrete hormones like insulin, glucagon, somatostatin, as well as other peptides like pancreatic polypeptide and ghrelin into the bloodstream. So what we are most interested in and is most important in digestion is the secretion of the exocrine pancreas. And the exocrine pancreas secretions are controlled by the vagus nerve as well as the hormones secretin and cholecystokinin, which are secreted by the enteroendocrine cells of the duodenum. So this image is a representation of a single acinus lined by those enzyme secreting acinar cells and its duct lined by those water and ion secreting centroacinar and ductal cells. So even though the ingested food encounters digestive enzymes in the mouth and in the stomach, it's the pancreatic enzymes that are the most important for digestion. So the pancreas really is the major digestive organ of the body. And these acinar cells, here's an electron microscopy image of an individual acinar cell and its zymogen granules at the apical membrane. But these acinar cells produce enzymes that are able to break down every class of macromolecule, carbohydrates, proteins, as well as lipids. And unlike the enzymes that are secreted in the saliva or in the stomach, these pancreatic enzymes are really vital for survival. So these enzymes are stored in the acinar cells in membrane-bound vesicles called zymogen granules. And each granule contains all different types of enzymes. And all of these enzymes are released when these zymogen granules fuse with the apical membrane of the acinar cell, releasing components into the lumen. So the proteolytic enzymes in particular are stored as inactive enzymes. Those are those enzymes that like trypsinogen or proelastase. They are stored in the inactive form to protect the pancreas from autodigestion, and then they're activated upon exposure to enterokinase and trypsin in the intestine. So the enzymes that are secreted by the pancreatic acinar cells are pancreatic amylase, and that's an alpha amylase that digests starch. There's the proteolytic enzymes that digest proteins. These zymogen granules also contain pancreatic lipases that digest lipids, and all of these enzymes, along with brush border enzymes that digest the small polysaccharides and small peptides, are all that's needed for the complete digestion of carbohydrates and proteins and lipids. So here we can look at the cellular mechanism specifically behind enzyme secretion by the acinar cells. So this group of acinar cells is secreting enzymes into this central lumen here. So this is the luminal membrane of those acinar cells, and this is the basolateral membrane of those acinar cells. And the duct is maybe way over here somewhere. So this group of acinar cells is secreting enzymes into this central lumen. They're carried through the ductal system, and then of course eventually pass through the sphincter vodi and into the duodenum. So if we look at the basolateral membrane and the receptors that are present, acetylcholine is the main stimulator of pancreatic enzyme secretion during the cephalic and gastric phases of digestion, while cholecystokinin stimulates enzyme release during the intestinal phase of digestion. So we look at the steps that occur in this process. First, binding of acetylcholine or cholecystokinin, depending on the phase of digestion, enhances production of IP3. This triggers intracellular calcium release, and elevated calcium causes these zymogen granules, which are present in the cytoplasm, to fuse with the apical membrane. This results in the release of all the digestive enzymes that are packaged within those granules into the lumen. At the same time, the calcium also opens these chloride channels on the apical membrane, and chloride is able to exit the cell. So this chloride exiting generates a transmembrane potential, with this luminal or apical side being a negative relative to the basolateral side because of this movement of chloride. This lumen negative potential then causes cations, primarily sodium because that's the major cation, to move paracellularly between the cells and into the lumen. So the movement of sodium chloride into the lumen creates an osmotic gradient. So it's this osmotic driving force for the accumulation of water in the lumen as well. So the net result is secretion of enzymes, of sodium chloride, and of water into the lumen. So if we move our attention from the pancreatic acinar cells and instead focus our attention on the pancreatic ductal cells, the main function of these ductal cells is to secrete a bicarbonate-rich fluid that alkalinizes and hydrates those enzymatic secretions of the acinar cells. So this aqueous component of the pancreatic juice that's secreted by the ductal cells is made up of water and the electrolytes, sodium, potassium, bicarbonate, and chloride. So after the pancreatic enzymes have been secreted into the lumen by the acinar cells, they have to be flushed along the duct to eventually be delivered to the duodenum. And so that's what these centroacinar and ductal cells do. So the electrolyte solution that they secrete suspends those digestive enzymes and provides the flow of fluid that's needed to carry the enzymes down the duct and into the duodenum. So now let's look more closely at the mechanism that causes secretion in these ductal cells. And the ductal cells respond to a really different set of inputs than the acinar cells. So over here we have the lumen with the flow going this way. So that means that the acinar cells are off the screen to the bottom somewhere, secreting their enzymes component into the lumen. And the ductal cells are secreting their aqueous and electrolyte component, and everything is flowing then down through the duct and eventually into the duodenum, which would be off the screen to the top. So the aqueous secretion by the ductal cells is primarily regulated by circulating secretin, and this is excitatory, as well as somatostatin, which is inhibitory. So the main secretions of these ductal cells are bicarbonate, and that's important for neutralizing the stomach acid as it enters the duodenum, as well as large amounts of water to wash the bicarbonate and enzymatic secretions down the duct and into the GI tract. So if we look at this process, the duct cells express high levels of the enzyme carbonic anhydrase, which allows the combination of carbon dioxide, which can be produced by local metabolism, with water. And that allows production of bicarbonate and a hydrogen ion through that intermediate carbonic acid. Then that hydrogen ion that's created can be secreted across the basolateral membrane in exchange for sodium using this sodium hydrogen exchanger. So this process prevents the buildup of acid from destroying the cell. So then on the luminal membrane, bicarbonate can be secreted in exchange for chloride, and this is via a bicarbonate chloride antiporter. So where does this chloride that is in the duct come from to be exchanged for the bicarbonate? Because remember, the bicarbonate is really the most important secretion here. So we need to have this chloride present so that this exchange can occur. And this chloride comes into the lumen through this CFTR transmembrane protein. So the majority of the chloride that is secreted through this CFTR channel is secreted in response to the hormone secretin. So when secretin binds to its basolateral receptor, it activates adenylyl cyclase, leading to increased cyclic AMP. And that cyclic AMP accumulation leads to opening of this CFTR chloride channel on the luminal membrane. And that allows the chloride to exit the cell. And the acetylcholine and cholecystokinin here are in parentheses because these things potentiate this process, allowing additional chloride channels to open. So the secretion of chloride into the lumen creates this negative transepithelial potential. So there's a lumen negative potential, and that drives the paracellular transport of cations. And remember, the primary cation is sodium. So it drives the secretion of sodium through paracellular mechanisms into the lumen. And now, of course, because we have this osmotic force, then water follows. The amount of water that accumulates is enough to carry the secretions of the duct and the acinar cells all the way to the sphincter of Oddi, where it then enters the intestine. If we look at inhibition of this process, somatostatin can bind to its basolateral receptors and lower cyclic AMP channels, thereby closing this CFTR chloride channels. And when somatostatin reduces or eliminates this chloride efflux, then there is less bicarbonate chloride exchange going on and ultimately less driving force for the accumulation of water. So the flow rate has an effect on the composition of the final pancreatic secretion, just as it does in saliva. When the pancreatic flow rate changes, the ionic composition of the final pancreatic secretion also changes. So in the pancreas, there's this reciprocal relationship between bicarbonate and chloride concentrations, and that's maintained by this chloride bicarbonate exchanger. So concentrations of sodium and potassium in pancreatic juice are isoosmolar to plasma, regardless of the flow rate. In the unstimulated state, when flow is low, the electrolyte composition of the pancreatic juice pretty closely resembles that of plasma, with a low bicarbonate and relatively high chloride. But then, as the gland is stimulated and flow increases, the exchange of chloride in the pancreatic juice for bicarbonate across the apical membrane of the duct cell produces a secretory product that is more alkaline with more bicarbonate and has a lower chloride concentration. So the high bicarbonate concentration upon stimulation is important because that's what acts to neutralize the acidity of the chyme that's delivered from the stomach into the duodenum. The pancreas also has some important endocrine functions. This group of cells is called the islets of Langerhans and contains cells that secrete various hormones into the bloodstream. So if we look more closely at all of these different cells in the islets of Langerhans, which I am outlining here, and the exocrine pancreas is all of these structures out here secreting into their lumen, and you can see that there are many blood vessels present as well. So if we look within the islet of Langerhans, there are alpha cells which secrete glucagon, and that glucagon increases serum glucose by increasing gluconeogenesis as well as glycogenolysis. There are also beta cells that secrete insulin, and that insulin can act on various tissues throughout the body to decrease glucose levels. Somatostatin is also secreted by the D cells in these islets of Langerhans, and it has inhibitory effects throughout the GI tract. Now we're going to look briefly at biliary secretion, and this topic is going to be covered in detail in the 4th and 5th weeks of the block, so we're really just going to do a brief overview. So the hepatocytes continually secrete the components of bile. So bile salts make up a large portion of the bile, and they're made from cholesterol. Other bile components include phospholipids, bile pigments, ions, and water. And bile travels through these bile ducts and into the gallbladder. The ductal cells that line these ducts secrete ions and water, just like we saw in the pancreas, and this secretion is stimulated also by secretin, just like in the pancreatic ductal cells. In the gallbladder, there are ions and water that are absorbed, and this concentrates the bile salts. Then, when the gallbladder contracts, bile is secreted through a ductal system, through the sphincter of Oddi, and into the lumen of the duodenum. And this is under the control of the hormone cholecystokinin. And cholecystokinin, or CCK, is the hormone that's secreted by the eye cells in the small intestine. So once this chyme reaches the small intestine, CCK is released from these intestinal enteroendocrine cells, travels through the bloodstream, and acts to contract the gallbladder as well as relax the sphincter of Oddi. And remember, cholecystokinin also stimulates enzymatic secretion from the pancreatic acinar cells. Then in the small intestine, the bile salts emulsify and solubilize the dietary lipids, allowing for digestion and absorption to occur. Then, when lipid absorption is complete, the bile salts return to the liver through this enterohepatic circulation. Bile salts are absorbed in the ileum and into the portal circulation, and they're delivered back to the liver, and those bile salts are extracted from the blood by the hepatocytes, and this recycling of bile salts reduces the need or the demand to make new bile salts. And we'll return to bile when we talk about lipid digestion and absorption as well.